Most couples reach a point in their lives where pregnancy is no longer a reasonable option and seek a permanent method of birth control that is more convenient and reliable than the methods that they may have already been using. Permanent sterilization of a male for purposes of birth control can be effected by bilateral ligation and severance of the ductus deferens, a procedure medically termed vasectomy. In this procedure, cutaneous incisions allow a urologist access to the spermatic cord as it ascends from the scrotum toward the inguinal canal. Each ductus deferens is carefully separated from the outer components of the spermatic cord and then tied and cut. Because some sperm may be beyond the point of ligation, a recently vasectomized male is advised that he is probably not sterile for several days following the procedure. Violation of this protocol has resulted in unplanned pregnancies.
Hi, I'm Mike Shively. Welcome to Introduction to Anatomy and Physiology. We've just concluded segments on the urinary system, and we're getting ready to talk about the male reproductive system. Anybody have any questions, though, anything that we've covered recently? Starley?
I know that when we ended last time, we were kind of going into uremia. Is that bad for you to have?
Uremia is high levels of urea in the bloodstream, urea being the end product of protein metabolism. And when that builds up to certain levels, it can start interfering with various body processes, especially central nervous system. It can be fatal, in fact, if the urea levels in the bloodstream get real high. So yeah, it can be bad. Of course, if you start approaching that, they'll put you on dialysis, you know, to reduce those if the kidneys are diseased that's causing the problem. Any other questions about anything? In fact, well, we're getting toward the end of the course here, and we've talked about almost all of the body systems. Matter of fact, depending on how you count them, we've basically done 10 body systems, and we only really have 2 to go, the male reproductive system and the female reproductive system. Or if you just think of those as the reproductive system, then we're going to talk about one more body system. But it is the only body system, I think we've mentioned before, where there's significant sexual dimorphism. In other words, we have different organs in the two sexes for the reproductive system. So we'll spend a session or two on the male reproductive system and then a session or two on the female reproductive system, beginning today then with the male reproductive system. Now, is this a visceral body system or a somatic body system?
Visceral.
Visceral. Tubular in design. Many of the organs are tubular structures, have openings to the outside, and there's a mucous membrane that lines most of those organs. So it's a visceral body system. Let's make a list of the components. And I'll tell you what, I'll put the first one on. Let's go to the overhead camera. We'll just write male reproductive system. And I'll write testis, which is Latin for testicle. And there are 2 of these. Now, I want you to, let's make this list in the order that we would encounter these things, say if we were a sperm. So testis is going to manufacture sperm. What would the next organ that sperm would encounter be? Seth?
The epididymis.
Epididymis. So we'll put down epididymis here. How many epididymides would there be in one human being, one male human being?
2.
2. All right, what would be next? Starley?
The ductus deferens.
Ductus deferens. Now, what is this often called? Instead of ductus deferens, it's often called what? Somebody, anybody?
Isn't it vas deferens?
Quite often in texts, physiology and anatomy texts, you'll see this called the vas deferens. I'm going to put that underneath ductus, and I'm going to write it in quotes here. When you actually look up this word, that means vessel, the deferential vessel, and it's really a duct rather than a vessel. So the proper name for the structure is ductus deferens. But the name vas deferens is quite often retained through the clinical procedure that was mentioned on that pre-taped video segment, a vasectomy. They still use the term vasectomy for ligation, bilateral ligation of this structure to effect, quote, permanent sterilization in males. All right, how many ductus deferens in a male would there be?
Two.
Two. So we'll put 2 back here on our list that we're making. 2 of these. And what would go next on the list? What would be next? Susan?
Urethra.
Urethra. Now I'm going to put urethra, if we go to the overhead camera, I'm going to put urethra in quotes, or not in quotes but in parentheses here, because the urethra really belongs to the urinary system. But it does have important functions in the male reproductive system. Now it's not going to be a part of the female reproductive system, but it does have a functional transport function purpose here in the male reproductive system. So we'll list it and we'll put parentheses around it just to remind us that it really belongs to another system that just has male reproductive functions. And then most of the urethra is surrounded by what?
The penis?
Bryce, you bet, penis. Now once we've said testis and epididymis, ductus, urethra, and penis, have we listed everything in the male reproductive system? We haven't, have we? We've got some other parts here. These are the structures that sperm would encounter, but we have some other structures. What are some of those other structures? Somebody, Seth?
The prostate.
Prostate. Now this is an organ that can either simply be called prostate or it can be called prostate gland, and both are correct. Prostate or prostate gland. And there's only one of those. We'll just leave that singular to imply that. But there are other glandular structures associated with the male reproductive system. Anybody know another one? Susan?
The seminal vesicle.
Seminal vesicles. Two of those. We'll leave that plural there, vesicles. And finally, there's another structure that in a lot of biology texts is called the Cowper's gland. Its official name, though, is what, Starley?
The bulbourethral gland?
Bulbourethral gland. There's 2 of those. All right, GL period. And well, I'll put the number here in front. 1 of these, 2 of these, 2 of these. And of course up here on the urethra and penis, there's only 1 of those apiece. So now we've got essentially everything in the male reproductive system accounted for. There is a structure, a spatial cutaneous pouch called the scrotum that actually houses testis and epididymis. And the ductus deferens goes down into it, but that really is just part of the integumentary system. It's just a skin pouch for the testis. So these are the major components. Let's compare the list that we just formulated to what we have up here on the graphic and see how we did. 2 testes, 2 epididymides. It's kind of hard to say in the plural form, isn't it? So we just said epididymis up here. 2 ductus deferens, a left one and a right one. Then we got the urethra itself functioning in the system, but not actually a part of the system from a home system standpoint. The penis surrounds most of the urethra, but not all of it. And then we've got 1 prostate and 2 seminal vesicles and 2 bulbourethral glands. Now, while we've got this graphic up, let's go back and take a look at each one of these things and kind of summarize overall what the functional purpose of each one of these parts happens to be in the male reproductive process. The testis. What's its job? Somebody, anybody, just blurt it out. What's it do?
Doesn't it make sperm?
Makes sperm, manufactures sperm. That's one of its jobs. So sperm are produced in the testis, and the testis does something else too. In addition to producing sperm, it has another secretory product. And this one is not a living cell, but what is it?
Does it produce a hormone?
Produces a hormone, produces testosterone. So the testis has kind of two jobs, sperm production and testosterone production. One of them is an exocrine function, and one of them is an endocrine type function, right? The testosterone is a hormone that goes into the bloodstream. That's endocrine. And the sperm are cells that are actually excreted through a duct system to the outside. That's the exocrine function. Okay, the epididymis. What is the functional purpose of the epididymis? What's it do? Tyler?
In the epididymis, the sperm mature.
Sperm undergo their final maturation. Now, they're manufactured in the testis, but they have to spend some time in the epididymis to complete the maturation process. And we're going to spend a little bit of time today on spermatogenesis, the process of sperm formation. So we'll come back to that and spend more time on that particular aspect. So they mature in the ductus deferens, or in the epididymis. Then the ductus deferens does what?
It's a transport.
Primarily, Susan, it's a transport tube. It's going to transport sperm from the epididymis into the urethra. Now, some sperm are, quote, stored there as well, but think of it primarily, the ductus deferens, as a transport tube. And then the urethra, of course, is a transport tube too. Now, most of the time, the urethra is when it's transporting something, that something is urine. Okay? Only during ejaculation are sperm normally found, ejaculation and nocturnal emissions are sperm normally found within the urethra. The penis, of course, is the male copulatory organ, and we'll talk about that a little bit more when we talk about human development. We'll talk about fertilization a little bit and what's involved there. Prostate and seminal vesicles. The prostate and the seminal vesicles both are producers of materials that are added to sperm to make semen. Let's go to just go to the overhead camera for just a second. If we start out with just sperm, and then we add the secretions of the prostate and seminal vesicles, then we have semen. So semen and sperm aren't quite the same thing. There's sperm within semen, but sperm are just the male sex cells. If you add the secretions of these accessory sex glands, sometimes they're called secondary sex glands or accessory sex glands, then the combination is called semen. This is the material that's ejaculated during orgasm in males. Okay? Then we left off, we haven't talked about the bulbourethral gland. That's the thing we said was formerly called Cowper's gland, and you'll still see that in a lot of biology texts. What's its job? What's this bulbourethral gland do?
Doesn't it just secrete lubricating?
The secretions of the bulbourethral gland, that's exactly right, Susan, are lubrications that are produced during sexual excitement in males, and the functional purpose is primarily to lubricate the glans penis of the male prior to intromission during sexual intercourse. So it's a lubricant producing. It precedes ejaculation, secretions of the bulbourethral gland. So those are the overall components. We'll actually look at these parts on some models and talk about them individually. But let's talk about the overall functions. Now, we said, you know, the testis did this and the ductus deferens does this and so forth and so on. But the system as a whole, the male reproductive system as a whole, well, produces male sex cells. We call those sperm. Each one's a spermatozoan, if you will. produces male sex hormone, testosterone, okay? Then combines the secretions of some of the accessory sex glands with those sperm to produce the fluid that we call semen. And finally, copulation or sexual intercourse is a function of the male reproductive system. So those are the overall functions. Let's take a look at a graphic to summarize those and make sure we got them down. Production of male sex hormone, testosterone, production of sperm, production of semen, that's sperm plus secretions of prostate and seminal vesicles, and finally, sexual intercourse. Any questions about overall parts and overall functions before we start looking at some of these on some models and talking about them in a little more detail? All right, let me bring a model up here, and we'll go to the overhead camera and look at this model on the overhead. This is a model through a more or less mid-sagittal plane, although we got some structures that are on this side of the mid-sagittal plane. Obviously, here's a testis down here within this cutaneous pouch called the scrotum. Now, why? Every male has asked this question. Why did God decide to put the testes down in a scrotum? Because they're relatively vulnerable, you know, when your gears slip on your bicycle or when you're high jumping a fence and don't quite make it, stuff like that. Then these things are down here where they can kind of get hung up on stuff, and it doesn't feel particularly good. There's a lot of sensory neurons associated with testes. Why are they down here? What's the purpose? Starley, what do you think?
For temperature? Like
Actually, it's a thermoregulation thing. You're exactly right. For some reason that's not clearly understood, males of most mammalian species can produce sperm only if the testes are a little bit lower than core body temperature. Now, the testes actually develop up in the abdominal cavity, and every once in a while a male is born that the testes refuse to migrate down. Heard about the bicycle crossbar thing or something, you know. But they refuse to migrate down into the scrotum, and those individuals are called cryptorchids. When the testes remain up in the abdominal cavity, the temperature just being a couple of 3 degrees higher is sufficient to interfere significantly with sperm production. And so quite typically those individuals are sterile because they simply cannot produce viable sperm with the testes being up there. Just by dropping the temperature just a little tiny bit, by having the testes down in the scrotum, the difference is significant enough to allow spermatogenesis to occur. Starling?
Is it possible surgically to bring those down?
Surgical techniques have been tried on cases where the testes fail to descend on their own. In general, most of those techniques have not been very successful. They've also tried some hormonal injections of growth hormone and that sort of thing, and that has had partial success in some cases. Now, sometimes we have a male that has one testis down in the scrotum and the other testis up in the abdominal cavity. Those males are usually quite fertile because one testis produces, you know, enough sperm for fertility purposes. Susan?
Are males cyclic like females are in their sperm?
No, once a male reaches sexual maturity, males produce sperm until the day they die in most cases. So there's no cycling or to the male reproduction. There's no cycle at all to the male reproduction. It's basically continual once a male reaches sexual maturity. At any rate, going back here, and we'll come back to this and talk about it some more. Here's the testis down in the scrotum. Here's the epididymis sitting on the side of it. Now, the epididymis is only about the same length as testis, you know, a couple of inches long maybe. And then leading out of the tail of the epididymis, and actually on this model you can't see much of it because they've got the nerves and blood vessels around it, but we've got the ductus deferens. Remember, this testis actually began development up here in the abdominal cavity. As a matter of fact, they start development right below the kidneys. And so during fetal development is when the testis and epididymis migrate down through the lower anterior body wall, and then finally end up, if everything happens normally, down here in the scrotum. Now, the ductus deferens winds around here, and actually I'm going to pull up a different model for just a moment. Here we've got one. This is the urinary bladder. Go back to that overhead camera for just a second. This is the urinary bladder, and here comes the ductus deferens, and it actually plugs in or passes through the prostate. So this bulbous enlargement here is the prostate. These structures on either side are the seminal vesicles. So they kind of sit on the side of the urinary bladder as well. Where that ductus deferens actually passes in to the through the prostate into the urethra. If we go back to this model now on the overhead camera, and follow me right through here. This is the ductus deferens coming in. This is the prostate right in here. And you see the ductus deferens actually just passes right through the prostate to enter the urethra. The enlargement of the ductus deferens at this location is called the ampulla of the ductus deferens. Not particularly important that you remember that. Excuse me, but the terminal part, where it gets kind of small here and actually empties into the urethra, is called the ejaculatory duct. And it is joined there by the duct from the ipsilateral seminal vesicle. So there's actually two structures that enter the urethra, if you will, where the ejaculatory duct comes in on each side. One of those structures would be the ductus deferens, and then the other would be the secretions coming in from the seminal vesicle on that side. The prostate surrounds the initial part of the urethra, and because of that, individuals that have prostatic hyperplasia or prostatic neoplasia, cancer, when we start getting excess tissue buildup right here, those males quite often have difficulty urinating because the enlargement of the prostate will then tend to choke off, if you will, the urethra that's passing through it. Now the urethra runs the full length of the penis. Here's the penis itself. Go back to, yeah, we're on this overhead camera. And it actually has a root which attaches to the pubis. It's got a body. And then the distal tip of the penis here is called the glans penis. So we got a root consisting of a left crus and a right crus, a body, and a glans penis. In an uncircumcised male, the tip of the glans penis here is covered with redundant skin. That redundant skin is called prepuce. And that's the skin that is removed, during circumcision. And one of the decisions that you will have to make as parents, if you haven't been there already, is whether you're going to have your little boy babies circumcised or not. There were some studies that were done a number of years ago that showed that wives of circumcised husbands had lower incidences of cervical cancer. But later analysis of that study has kind of shown that that study was a little bit stilted, if you will, because it was done in a certain ethnic group, and they've basically shown no difference in cervical cancer in women, whether their husband has been circumcised or not circumcised. I can tell you if the little boy babies had a vote, they would probably say, nah, no, we'll just not do that today. Kind of interesting. That circumcision, of course, has some religious overtones in certain religions, and so there are reasons, religious reasons for the procedure in some particular beliefs. But basically, in most of the Christian religions, that's 6 one, half a dozen the other. So it's not a requirement anymore like it was originally to become a member of the Christian church. You can imagine how tough it was to get converts back in those days. Oh, by the way, Susan?
I just had a question about the incidence of urinary tract infections in wives of circumcised versus uncircumcised?
You know,
men?
I don't know studies on that. One of the reasons, when you talk to the, you know, your neonatologist or your pediatrician about whether you're going to have your little boy baby circumcised or not, he'll basically give you both sides of the fence. And one of the things that he will say at that time is he'll say, well, it is easier to keep the glans penis clean without the redundant skin covering the glands. And if that's true, then it probably follows that there might be a little more problem with infections going on, you know, if in wives whose husbands are not circumcised. But any questions about that very quick run through of parts? I didn't show you the I did not show you the bulbourethral gland. Let me bring up another model because this one won't show it. Let me just kind of show you on this model, if we'll go to the overhead camera for just a second. Again, here's the urinary bladder. And here comes the urethra down out of the urinary bladder. This is the prostate right here surrounding the initial part of the urethra. The urethra comes on down. This little tiny glandular structure right here, there's a left one and a right one. And that glandular structure is the bulbourethral gland. And so its duct comes out and joins the urethra. Of course, this particular model's been more than circumcised. He's actually been transected right across here, because I'm missing that part. Oh, here he is. Here's the rest of him. There, I feel better now. All right. Okay. So there you kind of have it on a mid-sagittal section for this part. This structure right here, of course, is pubic symphysis region right here. So you can see the relationship of these various parts. And we're going to do actually some sectional views of the penis when we get there. I just want to kind of give you an overview of parts. And then we're going to start through these parts from the beginning, starting with the testis, and talk about the specific features of them and the physiology of what's going on there. Anybody have any questions, though, on that quick run through? We talked a little bit about parts and a little bit about functions. Any questions there? All right, let's talk about testis a little bit. And let's go to the overhead camera. And we'll say this is a testis, a couple inches long, roughly. Sitting on the side of it here is actually the epididymis. So we'll say that's the epididymis. And here goes the ductus deferens. So I'll just kind of color the epididymis black. Now, the outer part of the testis has a thick connective tissue capsule on it, and that is called the tunica albuginea of the testis. That actually translates white coat of the testis. So it's tunica albuginea of the testis. It's just, think of it as connective tissue capsule. So I'm just going to write CT proper here. If we were to section through the testis, and we'll draw part of it over here, not worry about getting the whole thing on the camera. But if we were to section through, of course, the tunica albuginea would be on the outside, and there are septi that extend into the testis that kind of divide the testis up into chunks, if you will, of functional tissue. So each one of these is a testicular septum, and this, and this, and this, and this, kind of reminding you in a way of the sections in an orange. Each one of these is a lobe of testicular tissue. Now, I want to go down and get kind of microscopic. If we If we could actually have a high-power microscope and look in here, we'd find that each lobe of testicular tissue is formed by hundreds/thousands of little tiny tubes that are called seminiferous tubules. And I'm going to erase this part of the drawing right here. And we'll just say this is a seminiferous tubule. Of course, we've got the magnification up big time. And there's lots of these within each one of these lobes. And all those seminiferous tubules come off and leave that central collecting area right there. That central collecting area in the middle of the testis is called the rete testis. So each one of the seminiferous tubules leaves and comes back to the rete testis. So this is a seminiferous tubule, and we've got it magnified many, many times. So within each lobule, of course, there'd be thousands of these things leaving and coming back to the rete testis, okay? What I want to do is I want to do a transverse section through one of these seminiferous tubules and magnify it a bunch so that we can actually get down and see individual cells within that seminiferous tubule. So take any tubule and do a transverse section you want, any place that you want, and let me erase everything now. And we'll put a circle here on this, on our drawing. We'll say that's a seminiferous tubule in cross section. Out here, near the outside of the tubule, are the stem cells for sperm, if you will. Those guys that go all the way around the outside, I'm not going to draw them all the way around the outside, but they go all the way around the outside. So think of each one of these as one cell, another cell, another cell. Those cells are called spermatogonia. Spermatogonia. These are male sex cells, great-great-grandfathers of sperm. Now, spermatogonia divide by regular cell division or mitosis, and when they divide, some of the daughter cells become other spermatogonia, and some of the daughter cells differentiate into primary spermatocytes. So I'm going to draw some red cells inside these. Of course, this would go all the way around as well. And those red cells are going to be primary spermatocytes. The cell division that produces primary spermatocytes is regular cell division. Spermatogonia, being typical body cells, are 2N. That means they have, for humans, 46 chromosomes, 23 pairs. Primary spermatocytes, since this was a mitotic division, primary spermatocytes also are 2N. They have 46 chromosomes. So I'll just put a couple of tick marks meaning the same thing here. Now, primary spermatocytes divide by the first meiotic division to form secondary spermatocytes. And I'm going to draw the secondary spermatocytes as green circles inside. Again, these go all the way around. I'm just not going to draw them all the way around. I know this green doesn't show up very well, but I think it shows up maybe well enough to see it. So secondary spermatocytes. And that was a meiotic division. The first meiotic division, if you remember from biology, reduces the chromosome number down to half. So the parent cells had 46 chromosomes. How many chromosomes are these secondary spermatocytes going to have? 23. These guys are in. They have 23 chromosomes. I'm going to write it, abbreviate it, chromos for chromosomes, okay? Now, the secondary spermatocytes divide by the second meiotic division. Remember, meiosis is a two-division process. There's a first meiotic division, then a second meiotic division. The second meiotic division doesn't change chromosome number, but it does take a double set of DNA in each chromosome down to a single set of DNA in each chromosome. And when they divide, the cells that they form, which I'm going to write here as little tiny black circles, these guys are called spermatids. And of course, they're N equal 23 again. Now, spermatids are not sperm, but they undergo a metamorphic change. They actually, you know how a caterpillar turns into a butterfly? Well, spermatids actually grow a tail and slim down and become sperm. So each spermatid forms one definitive sperm. Now, if we had drawn these all the way around, like they really are in real life, within a seminiferous tubule, what we end up with is a very small opening here in the center of the tubule with a bunch of sperm sitting right there, and their little tails kind of waving out in the middle of the lumen of that tubule. And then the other cells in this spermatogenic series of cells scattered, you know, toward the periphery in the order in which we've drawn them. Right on the outside, we'd have the spermatogonia, again, the stem cells. Inside those would be primary spermatocytes. Inside those would be secondary spermatocytes. Inside those would be spermatids. And then we'd have some sperm that are, just about ready to leave the testis. Now, we said a while ago that sperm don't complete their maturation in the testis. They have to go to the epididymis and spend some time in the epididymis to actually finish their maturation. But the total process, you know, from top to bottom, if we'd add sperm down here to our list, total process from here all the way down is called spermatogenesis. I'm just going to write it on the slant right here. Spermatogenesis, the formation of sperm. And this last step right here, going from spermatids down to sperm, that last step of spermatogenesis is called spermiogenesis. So that's just the metamorphic transformation of a spermatid to a sperm. There's no cellular division right here. Remember, this division right here was mitosis. The division right here between red and green was first meiotic division. The division right here between green and black was second meiotic division. And this was simply a metamorphic change. Again, the word for that metamorphic change is spermiogenesis. So spermatogenesis includes the last step, which we said was spermiogenesis. Now, once a male reaches sexual maturity, this process occurs continually at a relatively alarming rate, actually. It's amazing that we have time to do anything else. It can be affected by, states of health and that sort of thing, but males are usually active sperm producers in most cases until the day they die. Females, have a more limited reproductive life, and we'll talk about that when we talk about the female reproductive system a bit, but females normally, after, a bunch of years of fertility undergo a menopausal or change process where they no longer are actively ovulating and producing viable eggs for fertilization. At any rate, this is the general concept of what we'd find within a seminiferous tubule as far as the spermatogenic series of cells. Let's go back to that same overhead camera. I want to show you, of course we'd have another tubule right over here, another tubule right over here, and so forth. There's some cells out in here, in between adjacent seminiferous tubules, and those are called interstitial cells. It's all one word, but I was kind of pushing the margin there a little bit. So I'm going to hyphenate it, but it is all one word, interstitial cells. These sometimes used to be called, you'll see it in some texts, cells of Leydig. Interstitial cells of Leydig, L-E-Y-D-I-G, you'll sometimes see. Those are the cells, the ones that are located right in here. Those are the ones that produce testosterone. So the ones inside the seminiferous tubules, the actively dividing cells inside the seminiferous tubules, are the ones that are involved in sperm production, that whole spermatogenic series of cells that we just talked about. And the interstitial cells that are located between seminiferous tubules are the ones that produce testosterone. Now, in addition to these two groups of cells within the testis, there is a third type of cell that I want to talk about very briefly. And again, go back to the overhead camera for just a minute. Interspersed among these spermatogenic series of cells are some really big cells. They used to be called nurse cells or Sertoli cells. Normally, when you're looking through a microscope, you can't see them because there's so darn many of these spermatogenic cells that they kind of get clouded and kind of hide these other guys. But these cells the proper name for them is sustentacular cells. Kind of running out of room. I'll see if I can squeeze it in right here. Sustentacular cells. And the sustentacular cells are big cells that actually serve to metabolically and physically support the cells of the spermatogenic series. I haven't said this yet, but there are no blood vessels inside seminiferous tubules. There's not any capillary beds in here. There's capillary beds out in the interstitial tissue in between tubules, but we have no source of nutrition in here. And it's believed that the sustentacular cells absorb nutrients as necessary from the capillaries out here in the interstitial tissue, and then serve as a source of nutrition for these various spermatogenic cells as they're undergoing their various divisional processes. Not only do they metabolically support them in that way, but they also physically support them because these spermatogenic cells tend to be kind of physically embedded in the sides of those sustentacular cells, if you will, during the spermatogenesis process. So when we think of a testis, I want you to divide, the cells in a testis into three functional groups, if you will. Let's look at a graphic that will summarize this for us. Spermatogenic series is the spermatogonia, primary spermatocytes, secondary spermatocytes, spermatids, and sperm. Those are the ones that are involved in sperm production. The interstitial cells are the ones that are involved in testosterone production. And finally, these last ones that we talked about, the sustentacular cells, are the ones that physically and metabolically support the spermatogenic cells during the process of spermatogenesis. Is that clear? Three different groups. Starley?
So for one spermatogonia, you're going to get 8 spermatids. Is that accurate, or 4?
Should only be 4. Matter of fact, let's diagram that in a different way because it's a little bit confusing when we do it just with words. Let's go back to the overhead. Let's say this is a spermatogonium. And he's, of course, 2N. Oops. When it divides with the process of regular cell division or mitosis, let's just write that to remind ourselves this is regular division. Starley, some of these daughter cells become other spermatogonia, but some of the daughter cells differentiate into primary spermatocytes, okay? So we'll say that daughter cell became another spermatogonium, but this one went to med school, this one went to law school, okay? All right, so this will be a primary spermatocyte. Primary spermatocyte. That being the case, then this one will divide to form two secondaries, first meiotic division. And then each one of these will divide to form two spermatids, second meiotic division. And now, Starley, each one of these simply becomes one sperm. Okay, now, depending on how you talk about it, see, we usually start when we're talking about the actual spermatogenesis process for how many, we start with one of these, because some of these daughter cells up here just become more spermatogonia. Okay? So once you get a daughter cell that's going to differentiate into a primary spermatocyte, from that point on down, you're going to get 4 sperm out of each one of these. Technically, you don't get any sperm out of this guy until he divides again and forms, again, some primary spermatocytes. Does that make sense?
Does it ever get overrun with spermatogonium if some of them are just going back up to make more?
Well, stop and think. If some of these daughter cells did not become spermatogonia, then if all of them went into primary spermatocytes, if both of these became primary spermatocytes, the spermatogonia would be completely depleted after one division. You see what I mean?
Yeah.
So we have to keep replenishing the stock, the breeding stock, so to speak, of these male sex cells. So some of those daughter cells replace the spermatogonia that would be lost otherwise. Seth?
What's the timetable for spermatogenesis?
The total, in other words, how long does it take to go from up here clear down to here?
Right.
Is that the question? Weeks. It takes several weeks for the whole process within the testis. And then I don't know how long they have to spend within the epididymis, but it's some days in the epididymis normally. At least a bunch of hours in the epididymis. So it's, from the very beginning to the end. But we got a lot of cells that are in various stages all the time. I've read some figures on how many sperm a typical male produces in a minute or something like that. And it's an alarming figure because it's the whole process, the total ones that are rolling off the end of the assembly line is quite a large number. Any other questions about that process? Did that clarify the question you had, Starling?
Mm-hmm.
Any other questions about spermatogenesis or testes or anything like that? Bryce?
I had lived with a mission companion, and he said that he played rugby before he came on his mission. One of the guys he was playing with got hit real hard and exploded the testes. So does that just destroy the tunica albuginea?
It does. If you get a severe blow, into the groin, perineal region down there, you can damage the testes quite severely. Did he give you any more information?
Well, he said it exploded. He stuffed a handful of ice down his pants and kept playing. So,
Really?
so
I think I've seen that bumper sticker about what kind of testes it takes to play rugby, but I'm not sure if I've seen it.
what effect does that have on this?
I obviously don't know if, I imagine that's a case-by-case thing. It's like asking how much does a stereo cost, or if you have a car wreck, can you still drive your car? It depends, Sometimes it's just a scratch, and then sometimes, you get a hole in the radiator, and you better stay right there. So I don't know what all was involved, but I would imagine he was in, if he really did have that much trauma, I'm surprised he could still play. I'm surprised he'd still want to play, to be honest with you. So. Any other questions, Amber?
Does it ever happen that the primary spermatocytes, both of them, go back into the spermatogonia?
You know, that's a good question, and I don't know enough about the cytology of that whole spermatogenic series to understand, can you get two spermatogonia formed rather than some of them differentiating. I think what we need to think about is the spermatogonia are mitotically dividing, and some of the daughter cells just spin out and become primary spermatocytes, but I don't think it's necessarily, one of them does this and one of them does that every time. You may be, in fact, exactly right. Maybe sometimes when a spermatogonium divides, a given spermatogonium starly, maybe both cells become primary spermatocytes. I don't know. Yeah, question?
Sorry, just a quick clarification then. In the secondary spermatocyte and the spermatids both have just N. That's where they split, right? So it's the primary spermatocytes have 2N. And then
Yes.
every stage one of that's just N.
Let's go back to overhead for one more clarification on that. We got two meiotic divisions in spermatogenesis. First meiosis and the second meiotic division. In the first meiotic division, chromosome number is halved, just to kind of remind you in case it's been a while since you've had biology. So the first meiotic division actually reduces the chromosome number from 46 to 23, or from 23 pairs down to just 23 individual chromosomes. The second meiotic division does not change the chromosome number, because we have 23 chromosomes, but think of it as 23 doubles, if you will, because each chromosome has two sets of DNA. And one copy of the DNA from each of these 23 goes into one spermatid that's formed, and the other copy goes into the other spermatid. So the second meiotic division does not change the chromosome number. It simply reduces the amount of DNA within each chromosome. Does that make sense? And remember that the N number, it reflects how many chromosomes you got. For humans, 46 or 23 is diploid or haploid. Doesn't say anything about how much DNA is in each chromosome. Okay? Does that make that clear? Any other questions? All right, let's go on and talk about the epididymis a little bit. Let's go to the overhead camera and kind of, I know we put it on there a while ago, but here we got a testis with the epididymis sitting there in about this configuration, roughly. So this structure here is the epididymis, and this one is the testis. Now the epididymis has three parts rather. This part up here is called the head of the epididymis. This is the body of the epididymis, and this part down here is the tail of the epididymis. Then there's kind of a hairpin or umbrella handle turn, kind of a 180-degree turn. And in the anatomy lab, we say, okay, where it makes that 180-degree turn, then the tail of the epididymis is now joining the ductus deferens. So we'll use that turn down at the bottom as the dividing line between the tail of the epididymis and the ductus deferens. Inside the epididymis, there's a single tortuous duct. It's very, very tortuous and twisty. This epididymis is only, 4 or 5 centimeters long, but the duct within it could be 5 or 6 meters long. It's called, obviously, the duct of the epididymis. And every sperm has to travel down through the full length of the duct of the epididymis. And as it goes down through that very, very tortuous duct, it completes its maturation process. So remember, sperm are formed in the testis, but they complete their maturation in the epididymis, within that duct. Susan?
How do they travel to the top of the epididymis from
How
the
do they get
testis?
into the epididymis? That's a good question. I think we've got time to answer that. Let me erase that one and stay on the overhead. Let me show you what's going on. Remember me talking about a little central collecting area in here called the rete testis? And the seminiferous tubules all leave and come back to that rete testis. So that's that structure right there. Leading out of the rete testis are 13 to 15 little tiny microscopic ducts called efferential ducts. Let me write that down here for you. There's 13 to 15 of them. I'm not going to draw them all. And those efferential ducts go over here and actually plug into the head of the epididymis and carry those sperm then from the seminiferous tubules, which are looping out into the various testicular lobules and back, carry the newly formed sperm out into the duct of the epididymis. They go over here and join the head of the epididymis. So that's how those sperm get out of the testis into the epididymis. Okay?
Thanks.
So these efferential ducts are microscopic. You're not going to see those on a cadaver, but you can see them microscopically in histologic sections. Any other questions? Well, we've got just enough time maybe to talk about the ductus deferens itself. The main thing to remember is the transport tube. Joins the tail of the epididymis to the urethra. It passes through the prostate to get to the urethra, but it opens into the urethra. And certainly there's a number of sperm that are in it at any given time, so we could say that it, quote, stores sperm. However, a male that's not sexually active, the sperm are still being produced, and those sperm will eventually get shoved out of the male reproductive system by what are called nocturnal emissions, sometimes called wet dreams in the high school locker room. Okay, we'll continue with the male reproductive system next time.